Hello, my name is Madhav,
And my name is Sham.
and today we'll be presenting our work titled Computational Simulation of Progranulin for Blood-Based Detection of Parkinson's Disease up to 7 years before symptom onset.
So, firstly, we'd like to give you some background information about Parkinson's disease. On the left, you can see a chart detailing the progression of Parkinson's disease as a timeline, starting with 20 years before the clinical onset and going all the way to 20 years after the clinical onset. What's important to note here is the fact that most of the symptoms that are characteristic of neurodegenerative diseases mainly start after the clinical onset. On the right, you can see another chart that we made, which is a timeline of the history of Parkinson's disease, starting with Dr. James Parkinson's publication of an essay on the shaking palsy, which is the first publication of Parkinson's disease, and ending with the publication of the paper that our research is based on, which linked progranulin, which we will talk about shortly, to Parkinson's disease.
So, diagnosis of Parkinson's disease can be difficult due to its ambiguous symptoms. This means that, Parkinson's shares a lot of common symptoms with other neurodegenerative diseases, especially very early on. So, tests can be indicative of many different neurodegenerative diseases other than Parkinson's. But recently, Parkinson's has been linked to progranulin. So, higher progranulin levels in the bloodstream and brain have been associated with Parkinson's. And for some just quick basic facts on Parkinson's disease, there are around 200 new cases in the US every year. There's about 10 million cases worldwide. Some common symptoms include tremors, bradykinesia, slowed movement, speech changes, non-motor symptoms like depression, and more. And every 6 minutes, someone is diagnosed with Parkinson's disease.
So now we'd like to give you some background information about the protein that we're focusing on for our research, which is progranulin. Progranulin is a protein that normally regulates the cell cycle and inflammation and can normally be found in blood plasma and cerebrospinal fluid, or in the brain. Recently, through the research of Dr. Jenny Helkvist and her colleagues, the elevated levels of progranulin have been linked with Parkinson's disease. On the right, you can see a graphic detailing how elevated progranulin levels can end up leading to Parkinson's disease. So, progranulin normally binds to these five receptors that are shown. However, elevated progranulin levels can lead to overbinding of these, which can eventually lead to neuroinflammation and then Parkinson's disease.
So, aim and hypothesis. The objective of our research was to identify antibodies that bind to the progranulin protein for the early blood-based detection of Parkinson's disease, possibly enabling diagnosis up to 7 years before symptom onset. And we hypothesized that antibodies binding to the protein surface could be used to detect and diagnose Parkinson's disease.
So now we'd like to talk about our methodology throughout this research. The first thing we did was we retrieved the amino acid sequences of the granulin precursor, or progranulin, and the antibodies that we were going to focus on for our research from UniProt, which is a protein database that contains a lot of information about various proteins and nucleic acids. The second thing we did was we inputted these amino acid sequences into AlphaFold 3, a software that recently won a Nobel Prize for being able to predict the 3D structures of proteins simply by using their amino acid sequence. Finally, we predicted the potential binding sites on progranulin using a software known as P2Rank, which allows us to rank binding sites on a given protein based off the probability that they will be able to bind to.
After we predicted the potential binding sites, we then used HDoc, which is a web-based application that allowed us to perform docking simulations between progranulin and the different antibodies in our pool. We then used Prodigy in order to calculate the different binding energies so we could identify which of the antibodies binds the strongest with progranulin. Lastly, we used Chimera X in order to analyze the docked complexes and the electrostatic potential of progranulin.
After all the research was said and done, we needed to decide between the tested antibodies. To do this, we had two main criteria. The first was the binding energy of each antibody as calculated by Prodigy. Binding energy normally indicates how much energy is stored within a bond. However, in our case, most binding energy values are negative. This means that energy was actually released during the bond, making it more stable. Because a lower binding energy indicates a stabler bond, we want to see which one has the lowest binding energy because that will likely indicate which bond is the strongest. As such, we were looking for the lowest binding energies. The second criteria were the sites predicted by P2 rank. Although binding energy is an indication of how strong a bond actually is, it is pretty much irrelevant if the bond can't happen in the first place. This is where P2 rank comes in. P2 rank allows us to see the sites on progranulin that are most likely going to allow a bond to form, as well as the probability that a bond can form at that location. This, combined with the bonding simulations we got from HDoc, allow us a way to figure out how likely a given antibody is to bind to progranulin. With these two factors in mind, we are able to determine both how likely a bond is to form between an antibody and progranulin, as well as how strong that bond will be.
Now moving on to our results. So as you can see here, this is labeled figure 2, and figure 2 basically depicts all of the antibody and progranulin docked or bonded complexes. So as you can see, 1IGT is highlighted yellow because it's the winner, and it binds to the most optimal binding site on progranulin. And moving a bit to the right, there's a chart which shows the ranking of each binding sites. And next to that chart is a model of progranulin in white, which then has colored kind of blobs on them, which indicate just the best or the different binding sites or where the antibodies could potentially bond. So red is our number one spot, and pink is our worst spot. So 1IGT binds to this red spot, making it one of our best or our best choice.
Shown here is the second half of our research in the chart detailing all the binding energies in kcal per mole of each of the antibodies, as calculated using Prodigy. All the values here are shown here are negative, indicating that energy is being released in the bond rather than being stored. This indicates a stronger bond. As such, we are looking for a relatively low binding energy. However, it can't just be the lowest, as shown with 3GD6, because it also needs to have an ideal spot that is bonded to on P2 rank. Because of this, we chose 1IGT, as it has a very low binding energy while simultaneously also having a very good binding spot on progranulin, being the number one ranked spot. This means that it has a very likely chance of forming a bond, as well as that bond being very strong when it is formed.
Okay, so as you can see, we kind of came up with a pretty simple application test. So our application utilizes a rapid or antigen testing detection cassette, and we will be using the Fab region of the antibody or the fragment antigen binding part of the antibody, and that will be in our cassette first. Then we'll take the blood from someone that may or may not have Parkinson's disease, centrifuge it so we can separate the plasma and the blood cells. And then we're going to take a swab, dip it into the plasma, which the granulin precursor progranulin resides, add that to the detection cassette, wait for however much time may be needed for results to be shown. Then we'll search for the amount of granulin antibody complexes formed, and if there's an amount that's considered to be above normal, then that could potentially be indicative of Parkinson's disease.
Unfortunately, our research does have quite a few limitations due to its nature. Firstly, because the data was collected using existing datasets and AI powered computer simulations, we lack experimental validity of our selected antibody as we haven't actually tested it in a lab. Not only that, but because these are AI powered, they are subject to error, which means that potentially some of our research may be wrong. Secondly, only 10 antibodies were tested, which is a very limited sample size that may not capture the most effective antibodies for binding to progranulin. This means that because we have such a small sample size, we may have overlooked many antibodies that may actually be better than 1IGT for binding to progranulin. And finally, because of the lack of experimental testing, statistics such as the amount of time it takes to form a bond or for the bonding to be complete is actually unknown. And because of this, we don't actually know how efficient this diagnostic process will be.
Now for future work, which is a continuation of limitations essentially. So We need to first experimentally validate the selected antibody in a laboratory setting. So we can actually see if our research is viable in a real life setting. Then we would need to, or before that even, we would need to expand the study to include a larger pool of antibodies for better accuracy. As mentioned earlier, having more antibodies out there, because we only use 10 antibodies, there could be many more antibodies out there that are a stronger candidate than one IgT. Lastly, we would need to develop an optimized detection kit based on said selected antibody, as we use rapid or antigen testing, but there may be a more effective method.
Here are some of the references for our research. The main article I'd like to point out is the one by Dr. Jenny Halekvist and her colleagues called Plasma Proteomics Identify Biomarkers Predicting Parkinson's Disease Up to 7 Years Before Symptom Onset. This was essential in inspiring our research as it was the paper that originally identified progranulin as a biomarker of Parkinson's disease.
Thank you for listening to our presentation.